Professor Jae-Sung from Korea University, Seoul. Today's his talk is molecular mechanism underlying the fidelity of microRNA biogenesis. His great achievement is described on page 37 on abstract book. Professor Woo, please. 20 minutes lecture and 5 minutes discussion.
Thank you. Thanks to Professor Kim Hyo-soo and Dr. Kwon to invite me to this nice conference and organize all the other organizers of that. I thank you for. And I will talk about the molecular mechanism of microRNA biogenesis. It is detailed structure and molecular mechanism. But this could be important for designing a new method of microRNA therapeutics. So let me start it. So microRNA is 22 nucleotide RNA duplexes, which has characteristically 3' and 2 nucleotide overhang. This microRNA duplex can be transfected or it can be expressed in a cell, and then one strand is discarded and the other strand is loaded on the AGO protein. And this protein complex go to the target mRNA for the translational inhibition. The most important sequence is here from the second nucleotide to the 7th nucleotide. This sequence is called seed sequence. So seed match is important for targeting. Then how the microRNA is generated? Primary microRNA is transcribed by polymerase II, just like mRNA transcription. And this gene contains the stem loop structure, very long stem loop structure, and the microprocessor complex, and it contains the DROSA and DGCL8 proteins. This complex recognizes this structure and cut off the basal part. And another RNase 3 enzyme, like DROSA, and dicer cut off the apical part to generate the mature microRNA. There are various the microRNA therapeutics under development, and these therapeutics can be the microRNA mimic and pre-microRNA mimic, and also AGO microRNA complex. They can be transfected into the cell to inhibit certain gene. Or anti-microRNA can be also transfected into the cell to inhibit some functional microRNA. So there are several microRNA therapeutics in the clinical trial. They contain anti-microRNA and also microRNA 29 or 1634 mimics. So a few months ago, one siRNA drug, the first siRNA drug was approved in FDA. It is called Partisiran. As you can see, it's exactly this is a 21 nucleotide that is a modified duplex RNA. But there is another possible way to be the potential microRNA therapeutics using primary microRNA. So you can use primary microRNA gene in the normal vector, and this vector can be transfected to express primary microRNA. This will undergo the cellular processing pathway to generate functional microRNA. The advantage for this method is persistent expression and also tissue-specific expression using tissue-specific promoter. So we are interested in this process, the initial process, the cleavage by the DROSA and DGCLA complexes. And this step is particularly important because it determines microRNA sequencing. If if the DROSA process in a wrong site, for example, the one nucleotide shifted, then the seed sequence is also shifted to generate different sequence microRNA, and this will target different the mRNA pool. So the fidelity is crucial for the. We built the primary microRNA processing models in 2015, and in these models, DROSA interact with the two DGCLA molecule using C-terminal tail of DGCLA, and this C-terminal tail is important to stabilize, activate DROSA, and double-strand RNA binding domain enhances the efficiency, and the RHD domain enhances the accuracy. More importantly, DROSA can cleave, and also it can measure the distance from the basal junction. It's about 11 base pair. So we also determined the crystal structure of DROSA, and based on the structure, we specified much more the microprocessing model. By this model, we now understand where are all the sequences and where are all the domains and functional domains relative to the sequences. But it is still unclear how the cleavage site is determined by this the complex. One could easily imagine because the double-strand RNA is stiff, microprocessor probably recognize some motif and measure the distance to find the cleavage site. However, there are many primary microRNA features. First of all, the structural motif is the stem length is about 35 base pair, and there is always an apical junction and basal junction here. And there are many sequence motifs have been found: UGU, MGHG, NUG, and CNNC motif. And these motifs has a certain distance from the cleavage site. So at least 5 motifs: the apical junction, basal junction, and UGU, UG, GHG motif. And these are in a certain distance from the cleavage site, and they can increase processing efficiency, so likely have an impact on the cleavage site selection. But it is still mystery how the cleavage site is determined, because actually endogenous primary microRNA has all the distance which is different from each other. So for example, this primary RES7 family, here is the lower stem, which is from basal junction to the cleavage site, is sometimes 14 base pair, sometimes 13 base pair. And the apical junction is from the 21 base pair to 23 base pair. They are various. And in other primary microRNA, about 200 primary microRNA have tested, and many have 13 lower stem lengths, but still 14 and 12 base pairs are found. And also many primary microRNA have 21 and 23 upper stem lengths. So it seems to be clear the cleavage site is selected by these 5 different motifs, but the distance is different from each other. So how can we determine? How can you find the cleavage site determination mechanism? So the most important thing is what is more important than other motifs. And if we can measure the relative strength of these 5 motifs. And this is a complicated problem, but moreover, the GHG motif is not simple motif. And GHG motif was initially found by the high-throughput study. And then from -7 to -5 from the cleavage site. And these 6 nucleotides was randomized, and they made about 4,000 variants, performed the in vitro processing and measured the cleavage efficiency and to define a GHG motif. But this efficiency can be also presented as MGHG score. As you can see, his many sequences which is not included in the GHG motif still has a high efficiency. So it is more reasonable to use MGHG score rather than binary definition of GHG or the rest. Then this is more complicated, and probably high score MGHG will have higher effects on the cleavage site determination. And also there are 4 different the motifs, and lower score will have a smaller effect. So how can we solve the measure the relative strength in this complicated mechanism? The Sung-Chul, who led this project, one day he came to me with a big smile and he showed this substrate design. And this was a quite brilliant design. First of all, it has 6 UA base pair here. And what this means is here is 4 boxes will have the same MGHG scores. So here, so all 4 boxes have the same score. Then this will remove positional specificity. And GHG, a high score GHG motif can be included, and medium and lower included. So we can compare these 4 sequences. And also, this part and this part are around the junction are randomized. The sequence B indicate U or G and C, and M indicated A or C. What could be the variables in this artificial substrate? It will have 4 variables in GHG motif, and also this part will make or remove a UG motif, and also location of UG motif will change, and location of basal junction also can change. If you have, if this have new matches here, it will have a longer stem length. And the other one, the other part is also have, can have or don't have UG motif, and also the location of an apical junction will change. So there will be competitions and cooperation of 5 motifs. If these motifs indicate pinpoint the same cleavage site, then they will cooperate. And if they compete with each other, for example, one has certain pinpoint this cleavage site, the other has this cleavage site, they will compete with each other. So, and we designed the high throughput experiment. And we used 3 different backbone. This part is different. And MGH variant, I showed 4 different variants. And also random sequences, the basal part and apical part. And we performed in vitro processing using recombinant Drosha and digicellate complex. And we sequenced all the variants and 5 fragments of all the variants, and cleavage position and efficiency was measured. And the result was quite surprising. First of all, the 6U sequence actually worked. Here is we measured the 6 different cleavage sites. Here, the 6 different cleavage sites, and each line indicate the primary microRNA variant is about 500 variants here, and relative cleavage is presented as color. So the dark colors mean the more cleaved position. So if I zoom up here, and the 6U sequence, as you can see, they are cleavage sites are heterogeneous. It's because UG or AJ, the other sequence motifs or the structural motif actually contribute to the cleavage site determination, not because the GHG motif is not there. But we included the GHG, a high score GHG motif, and it showed extremely homogeneous pattern. Even the lower scores, like 65 scores, it showed almost homogeneous pattern here, which means high score GHG motif will is the champion. It actually win the other the motifs. But it has a lower score than 35, then it's now the cleavage pattern is heterogeneous again. That means this lower GHG motif is weaker or the comparable efficiency with the other motifs. Then how is the contribution of other motifs? We showed we separated this data into the 4 groups. The first group has only apical junction and basal junction, but UG and UG motif are not there. And the second one has UG motif there, and third one has only UGU motif there, and fourth one has all the motifs. In this group, basal junction always win the apical junction. And here the basal junction and UG has a comparable effect. And here only basal junction win. And here is also basal junction and UG have a comparable effect. So the basal junction and UG are important, but AJ and UGU are much less important. And what does the BJ and UG, the comparable efficiency of BJ and UG mean then? It means, I will give you an example of here's the UG motif pinpoint the C0 cleavage site, and the basal junction indicate the 13, the C1 position here. So in this case, C0 and C1 cleavage site are both active. So we can measure the comparable amount of cleavage on both sides. And what about the UG and how effective is the cooperation of UG and BJ if we can depict the GHG motif? So if you see the GHG sum as the 35 data, and here you can see the heterogeneous pattern. This is from the UG and BJ cooperation. So the GHG 35 has equal the efficiency with the UG and BJ cooperation. So we concluded cleavage size is determined by the MGHG motif, first of all, and the basal junction and UG motif. They will compete and cooperate with each other to determine the cleavage site. Then how the GHG motif is recognized by DROSHA. In our structural models, DSRBD is close to the GHG motif, so we performed the mutational study, we made also model the recognition model based on the structure of a bacterial RNA-3 protein, and we performed the mutational study and found that the mutations on these sequences show the defective recognition, so they didn't respond to the MJHG motif movement. So I skipped the experiment because of the time limit, and I will show you our model for summary. This is structure of DROSA, and this is primary microRNA. And initially, primary microRNA is recognized by the DDCL8, and it tried to fit with the central domain of DROSA. And DSRBD at this moment is slightly far from the RNA, but it will have conformational change to recognize the GHG motif. And then it finds the best position then the cleavage is now completed. Oops. So this work was mostly done by the Sung-Chul, and we for about 5 years we collaborated with the Larry Kim group in IBS, and we produced the protein sample, and the Seung-Kil helped a lot for protein production and the biochemical studies. And I thank all these contributors, and also thank you for your attention.